Thanks for inviting me, and also thanks for scheduling my talk at a reasonable hour. It's 9 o'clock Singapore time, and I've had numerous uh, meetings and presentations at 2 or 3 in the morning since this COVID thing started. And I have to say that I'd rather travel to the site than give the talks at 2 or 3 in the morning. I think probably the listeners would agree with that. So hopefully at 9 I'm still coherent. And let me pull up my slides and get started. All right, so thanks again for inviting me. I'm going to tell you a little bit about aging today, and I'll start with Singapore because that's where I am. But what I'm telling you is happening at various paces throughout the world in every country. In Singapore, we're looking at a situation where 1 in 5 Singaporeans are going to be over the age of 65 by 2030, and with the low birth rate that's also happening in the country simultaneously, what that means is we're only going to have 2 people working for every retired person. So the Singapore government is very progressive and has tried to identify problems before they happen and try to take progressive approaches to deal with them. And so they've identified aging as a major concern for the population and recognized the need to support the aging population. Just to give you an idea of what this looks like, just like everywhere else in the world, here on the left, you can see that while life expectancy is going up, Singapore is one of the longest lived countries in the world. The unhealthy years of life are also going up. And so we're keeping people alive longer, but we're keeping people sick longer. And I think this statistic here on the right is really fascinating. What it indicates is looking at people over the age of 65 with no chronic illnesses, 1 chronic illness, or 2 or more chronic illnesses. And this is the cost in Singapore dollars. 1 Singapore dollar is about 70 US cents, just to give you an idea. And the Singapore has a very efficient and effective healthcare system, so costs are much lower, but you can see that there's a 6-fold increase in cost as you get chronic diseases. So I think this illustrates the point that if we can keep people healthy and keep these numbers down around here, there's a massive savings to the government and the healthcare system in Singapore. In fact, we've estimated through our collaborators that a 5-year increase in health span would result in about $3 billion in savings. Now, this has been a slide that Matt Kaberlein and I have been using for a long time now. And I think it illustrates the whole point of the Geroscience initiative and the impact of aging research, and that previously and still many clinicians and researchers consider all of these diseases as single entities that don't interact with each other. And so you spend a lot of money trying to understand one disease at a time and develop treatments for that disease. And I think what the aging field has recognized a while ago now, and I think most people agree with this now, is that there is something in common. In fact, the biggest risk factor for all of these diseases and many other chronic diseases as well is aging itself. And so I've had made a pact that I'm going to stop using this slide because I think everyone in the world's seen it and I'm tired of talking about it. But the COVID-19 thing also made me rethink, and it made me consider the idea that even though we're talking about all of these major diseases, we're probably underestimating the potential impact of slowing down aging. And that if you look at aging, it's not just driving chronic diseases. As we know, and many of you know, it's driving functional decline as well. And what's been driven home to us this year, even though it was apparent before, is that it mediates the severity of acute diseases. So you're about 8-fold more likely to die from COVID-19 if you're over the age of 65. And that's not just true of COVID-19, but it's true of influenza and many other acute diseases that occur in the elderly. So slowing aging is going to have an even bigger impact than preventing chronic diseases. It will reduce functional decline and keep people from dying when the next viral outbreak happens. I'm not going to talk too much about mechanisms today, although I will touch on it in various places. But I do want to make the point that a number of these papers have come out talking about hallmarks or pillars of aging. This is the one that came out from the Geroscience initiative from the NIH, and a number of professors were involved in this. 7 different components were identified as pathways that modulate the aging process. But I think as I and others helped write this review, what we realized is that these things are all connected to each other. It's very easy in the literature to go from any component to another. So you can go from adaptation of stress to metabolism to inflammation to epigenetic changes to stem cells, etc. And so I think when I think of healthy aging, I think about maintaining this homeostatic network. So damage is happening to your body as you get older, but your body compensates for that damage and you retain your functional capacity and you retain your ability to sort of maintain a healthy equilibrium so you don't get chronic diseases. And it's when this network breakdown occurs at enough nodes that this whole thing falls apart. And that's when you get chronic diseases. And depending on how you spin out of equilibrium, that may define the different diseases that you come down with. And I think what this also suggests is that there's not really one cause of aging, but you can initiate aging or accelerate aging by targeting any of these nodes and potentially others that we haven't identified. And that different people may have accelerated pathways that are the same or different from other people. So aging is going to be very personalized, and I'll come back to that when we talk about biomarkers a little bit later. Now, none of this matters. I can talk about aging as the biggest driver of disease, but it doesn't really matter unless we can develop interventions that slow down the aging process and mitigate these diseases and maintain function. And this was something we developed with the company Syntara, looking at potential interventions to target different components of aging. And I think it illustrates generally what's been developed. The first thing that was really developed are lifestyle interventions, exercise, diet, stress management, mindfulness. These things are all in some stage of development and are all argued to slow down the aging process. And I think in some cases, like exercise, there's very good evidence, and also in diet. Now I think we're in this second phase where we're looking at drugs and natural products that can slow the aging process. And I think this is a critical phase as we develop translation to target aging. We have to show that some of the interventions that have been developed in this space that are effective in animal models will translate to humans. And I'll come back and talk about that down the road. And then there are much more radical interventions that are being developed as well through regenerative medicine, through gene therapies and other things that may not be quite yet ready for clinical testing, but show great promise in the long term. So we're moving outward in this circle as we go down the path. And I think that it's critical now to show that this second component of the circle can produce validated interventions that affect aging. Now, there's no shortage of potential interventions in animal models. These are just ones that I work on in my lab. And I'm not going to go through them, all of them today. If you have particular interests in specific ones, I'm happy to talk about that later. And we, as you may know, have done a tremendous amount of work on rapamycin and the TOR pathway. But today I'm going to talk about two other ones, alpha-ketoglutarate and Spermidine. And I'll start with alpha-ketoglutarate. And just as a disclosure, this is a company, the Ponce de Leon Health, that sponsored the research that was performed by Gordon Lithgow at the Buck and also in my lab. We did many of the mouse studies, and Gordon did many of the worm studies and some of the mouse studies as well. And the idea here is to look for combinations of natural products that have additive effects on lifespan and health span. And this is for two reasons, really. One is that we believe these combinations are likely to have synergistic effects. And second of all, from a company perspective, if you want to generate IP around products, you need combinations of natural products or specific formulations that produce unique results in order to get that IP. And so we've been looking at these kinds of combinations. I'm not going to take you through all the combination data today. I'm really going to focus on the one molecule that jumped out at us as something that's really impacting the aging process, and that's alpha-ketoglutarate. I said disclosure and then I didn't disclose. I'm actually a board member and I do research for PDL Health. So this is the structure of alpha-ketoglutarate. I'll come back to it in terms of what it does to the cell a little bit later. We decided to test it among about 30 or 40 other natural products reported to affect aging because of this paper in 2014, which showed that it could extend lifespan in worms. And Gordon's lab was able to repeat these findings very robustly. And so we started studying them in mice. And this is a data I'm going to show you has been accepted in Cell Metabolism and will be coming out soon. What we did is initiate treatment with alpha-ketoglutarate at 18 months of age in both male and female mice. And if I just showed you this slide, you might not be that excited because the effects on lifespan are modest, about 10% in females and about 5% in males. It's significant in females. It doesn't quite reach significance in males. We've repeated the study again and found almost identical results. If you combine the two studies, which you're not really supposed to do, but I'll tell you anyway, then you reach significance in males. But the effect on lifespan is not what got us the most excited. What really got us excited was the effect on health span. And to do that, we monitored frailty. And so we used this 31-point frailty index in mice that was developed to be relatively non-invasive and to be analogous to, as far as possible, to frailty indices in humans. And this was developed by Susan Howlett in Canada. And so we have started using this frailty index in all of our experiments on aging because we like it so much. So I'll just take you through this slide. On the left, you're looking at each dot as an animal at a different age, 18 months all the way up to 31 months. And this is a frailty score. And you can see that the control animals in blue, the frailty goes up faster than the AKG treated animals in, I guess, magenta. But this is kind of a misleading statistic because what's happening is animals are dropping out of the statistic because they're dying during this window. And so in other words, if you take this animal here that had a very high frailty score at 23 months, it doesn't show up at 25 months because it died in the interim. And so to show this data more clearly, what we did is we regraphed the data after all the animals died in terms of the percent of the animal's lifespan when we took the measure. So in other words, all of these points occur within 50 and 60% of the lifespan of the animal, 60 and 70%, 70 and 80, etc. And what you can see is in the control animals, frailty goes up pretty steadily over the course of age. But in the AKG animals, it's largely blunted. In fact, what we see is a 50% reduction in frailty in the female animals. I'm not going to show you the males, but we see almost the same effect. It's about a 40% reduction in frailty. And that's led us to conclude that AKG is having the effect of compressing morbidity in these animals. So it's extending lifespan, but it's extending health span and reducing frailty much more dramatically. The animals stay healthy for a very long time, and then they decline very rapidly at the end. Here's another way of presenting the data. And here what we've done is ignored the animals, the longitudinal effect of the study, and just plotted the frailty score versus the remaining lifespan, either for control animals or for AKG treated animals. And interestingly, if you look at the females and the males, frailty scores in control animals predict the remaining lifespan of the animals. So you can see a very good p-value for both females and males. But if you look at the lines in the magenta, there's almost no slope to those lines. And in fact, the frailty score very minimally predicts frailty in females and doesn't predict frailty in the males. So what that again suggests is that these animals are very protected against frailty, even though they eventually die slightly after the control animals. Just to show you the animals themselves, this is 27 months of age. Control animals are having all kinds of problems. Loss of hair, hair graying, although that's a little bit hard to see. Coat condition is dramatically impaired. You can see other kinds of problems in different animals. The IKG treated animals are largely protected from especially the frailty score, the frailty components that relate to coat condition. Their coat remains much better during this period of time. They're also more active. And what we've been trying to understand mechanism, and one of the things I'll point out, there's a lot more data in the paper, is that AKG seems to enhance the stem cell function in the skin or in the hair bulbs. So melanocytes are preserved with AKG-fed animals in green, although they're lost by this time point in the control animals, accounting for changes in hair color. We also believe that other adult stem cell populations are likely to be preserved in AKG-treated animals as well. we're looking at this in muscle stem cells and tracheal stem cells at the moment. And this may be one common feature by which AKG protects animals with aging. Now, if you go back to this molecule, this is a very well-known molecule, alpha-ketoglutarate. All of you that have had a biochemistry class have talked about it. It's in the middle of the TCA or citric acid cycle, so in central metabolism. It's important for converting glucose into components that can feed into mitochondrial pathways and generate energy. But I should also point out that alpha-ketoglutarate is involved in a large range of enzymatic reactions in the cell. It's involved in amino acid synthesis and probably is a substrate or cofactor for hundreds of different enzymes in the cell. It's a little bit like NAD, and many of you have thought about NAD in aging, is that this molecule is doing so many different things in the cell that it's a little bit hard to understand which of those might be beneficial for aging. And so we see a lot of benefits when we administer AKG. We see preserved adult stem cell function. We see better metabolism, and I would say that as metabolic flexibility. One reason I say that is AKG is a central component converting amino acid catabolism and carbohydrate metabolism, so it helps cells convert back and forth between different energy sources, and that seems to be preserved with aging. We see enhanced respiration, altered microbiome function. This is something we're studying in great detail, in more detail, and then a dramatic effect on inflammation, and I'll show that to you in a minute. In addition, we're starting to see changes in aging biomarkers, including in humans that are taking the product produced by PDL and reporting their epigenetic clock scores back to us. I'll come back to that later. So there are a lot of different components and pathways of aging that are affected by AKG. This inflammatory component is quite dramatic. These are different inflammatory cytokines here. You can see that from 18 months to 28 months, many of these boxes become more red in the control animals, indicating higher expression of inflammatory factors, and much of that is blunted with the AKG-treated animals. You can integrate the scores of all these different components and come up with a general inflammatory cytokine index and show that that's dramatically reduced by alpha-ketoglutarate treatment. I won't go into too much more detail on that, but this may be one of the central pathways that's being affected by AKG, although we don't yet know how. Now, I want to come back to something that more recently we're studying, and this is data that's not yet going to be published, but we were interested by the fact that alpha-ketoglutarate has been widely used to enhance exercise performance. I just pulled out 3 things off the internet here, showing suggesting increased athletic performance, better muscle recovery from wounds and surgery, and also improved bodybuilding and other components of exercise. And one of the things that people taking AKG have reported to us is they see improved exercise performance. And often AKG is given with arginine and other amino acids. So we're not doing that. We don't necessarily recommend supplementing amino acids. We're just looking at alpha-ketoglutarate by itself. But we wanted to look into this in more detail because the mechanism is not entirely clear for what might be happening. And one thing that jumped out at us, and the reason we got interested in this, I should say, is that in our studies, we were finding that AKG is not really taken up by cells at a very high level in culture. When we give it to animals, we can see that it gets taken up in the intestine, goes in the bloodstream, gets to the liver, but many other cell types don't seem to take it up. But in looking back at the literature, which is always a good idea, my postdoc, Guan Choping, found in 1963 that AKG was reported to be utilized by red blood cells for glutathione synthesis. This is actually before I was born. I'm not quite that old yet. And so we've been looking into this in somewhat more detail. Now, red blood cells don't really have mitochondria, so making glutathione and many other molecules has to occur through different pathways. And this has been studied more as the time has moved on. And so one study, in fact, found that 89% of the glutathione in red blood cells in tracing studies was derived from alpha-ketoglutarate. And this is a mechanism by which glutathione is made. It's basically a combination of 3 different amino acids, and alpha-ketoglutarate is a key component of glutamate synthesis, which is one of those components. So red blood cells take up AKG quite dramatically. If you look at our data, what we found is either in vitro, in cell culture, administering AKG to red blood cells, you can get a pretty dramatic increase in glutathione synthesis, or in vivo, although this is very preliminary data, we can see increased glutathione levels in red blood cells when we administer AKG. We would have a lot more data on this right now, but we've kind of got halted by COVID-19, so we're just getting back to these studies right now to try to confirm this finding. It's a little early. But when you take these components and together, this should be glutathione, decreases with age in red blood cells. Red blood cells readily take up AKG, driving glutathione production. Red blood cells are particularly sensitive to lack of glutathione because remember, they're carrying millions of oxygen molecules around with hemoglobin, and so they're sensitive to reactive oxygen damage, and even hemoglobin is sensitive. And so what we think is one particular protective mechanism of alpha-ketoglutarate with age is that it's restoring glutathione, increasing red blood cell function, and probably assisting oxygen delivery to muscle, which can affect both exercise and probably help people as they get older and start to have reduced muscle function. So we're very interested in this pathway right now and looking at it in some detail. I should point out that increasing oxygen delivery to peripheral tissues might be very beneficial in the context of COVID-19 as well, in patients that are suffering from acute respiratory distress syndrome. So we've given some thought to whether it might be useful acutely to help people get through that very late stages of the virus. So knowing this, we've now started two human clinical studies. One is being run by the company, PDL Health, and it's being sponsored by the company, but run by Indiana University, looking at the actual product, which is an AKG plus other components for male and female. So we're looking at the product using placebo-controlled studies, 45 to 65-year-old patients or participants that are generally healthy. We're like 100 total, 9-month intervention, confirming safety. Everything in these components is reported to be generally regarded as safe, but we want to confirm that. And particularly for outcomes, what we're looking at is inflammatory markers and also aging biomarkers. And I'll come back to that as an outcome for aging intervention studies. And then at NUS, we're doing a study through our academic approach, just looking at the sustained release calcium AKG without the other components, because we want to try to get to the mechanistic role that AKG is playing. But otherwise, it's very similar, 6-month study, similar participants, although there'll be Chinese ethnicity, placebo control with 2 doses of AKG, and again, looking at immune and markers and aging biomarkers, but also metabolic markers and other physiologic parameters. So we're hoping that these studies will yield data suggesting that the effects in mice can be preserved in humans. Now, I want to turn to Spermidine. And we got interested in Spermidine because of a lot of work in the aging field. Frank Medeo, Guido Kramer, and others have shown many beneficial effects of Spermidine. And it's a lot like other molecules that have been reported to affect aging, that it seems to do everything. It can have all kinds of different effects in different tissues. And we wanted to understand it a little bit better in experiments in our own lab. It can be delivered orally, and here's different foods that have high levels. It can be produced in the microbiome, microbiota in the intestine, and it also can be produced by cellular metabolism. And one of the interesting things to us was that even though it can extend lifespan, and this is 2 studies here that have shown that, it has a modest effect on lifespan extension when delivered in the drinking water, starting at early age, both in females. Well, here's the female data and the female plus male data. The surprising thing, though, was that it was not reported to have large effects on metabolism. And we were, most aging interventions do positively or have some beneficial effect on metabolism. So we wanted to understand what Spermidine might be doing. Here's the initial report showing that no effects on body weight, no effects on lean mass, no effects on fat mass, and they did a number of other studies showing the very minimal effects of Spermidine. So we wanted to go back and look at metabolism in a little bit more detail, and we did our study slightly differently. We started 3 different kinds of interventions with Spermidine, and I'll tell you those in a minute. We did not set out to do lifespan studies, so don't beat me up over these very small numbers, but I just put this up to show that we did keep some animals alive until they died of old age, and what we found is at least consistent with the idea that spermidine affects lifespan. So we seem to be having a similar effect to what's being published, but that wasn't the primary goal of this study. So what we really wanted to do was look at the effects on metabolism. So we started either young mice on spermidine, old mice, or young mice on a high-fat diet. And to make the story short, young mice, we saw almost no changes with spermidine. But when we put young mice on a high-fat diet, we saw dramatic differences. And also we saw some effects in old mice as well. But to focus on the young mice for a second, what we found is they were protected from body weight gain, and they're protected from increase in fat content over time with spermidine. And this is quite a dramatic effect. And if you go look at this in more detail, using metabolic cages, the young mice really don't show any differences across these 4 different measures. The young mice on a high-fat diet, however, showed increased energy expenditure, increased wheel meter running, and also increased wheel meter running. And we also saw benefits in the old mice, both in increased energy expenditure, increased wheel meter running, and increased wheel speed running in the metabolic cage. So both the old mice and the young mice show beneficial effects of the young mice on a high-fat diet and the old mice show beneficial effects of spermidine treatment. We've been trying to look at particular mechanisms for this, and one thing that's jumped out at us is that in visceral fat, there's a dramatic increase in expression of factors involved in lipolysis, which could account for the breakdown of fat and the protection against the increase in fat in a high-fat diet. So this is, you can see that spermidine increases expression of ATGL, HSL as well. And we think this means there's an increased triglyceride breakdown in visceral fat. No effect in subcutaneous fat, interestingly. And we see pretty big differences between subcutaneous and visceral fat with spermidine treatment. One other thing that jumped out at us is that spermidine fairly dramatically increases expression of FGF21. You can see that in the liver of old mice and in both the liver and the subcutaneous fat, interestingly, and not the visceral fat of the young mice on a high-fat diet. And given the protective roles reported for FGF21 and the fact that FGF21 transgenic mice have a long lifespan, we thought this might account for some of the longevity benefits of spermidine. However, when we got FGF21 knockout mice, we didn't do lifespan studies, but were able to show that these mice still maintained they were protected from body weight gain and fat mass gain on a high-fat diet. So FGF21 alone is not required for the beneficial effects of spermidine. Now, the other obvious candidate was autophagy. And so we also tried to look a little bit at autophagy, and we can see increased autophagy in some tissues with spermidine treatment consistent with previous reports. And so we went back and we looked at a fat-specific knockout of autophagy. And at least in the fat tissue where we're seeing the effects on lipolysis, we don't see a requirement for autophagy. So again, the mice are still protected from body weight gain and fat increase. So this suggests one of 2 things, either that it's autophagy in other tissues that's mediating the effects of spermidine, and that's causing a non-cell autonomous effect on fat tissue, or that it's a combination of different pathways, maybe autophagy and FGF21 and or something else that's mediating the benefit. So we're still trying to better understand the mechanism by which spermidine is beneficial, but I can say that we can repeat, at least we strongly believe we can repeat the longevity effects of spermidine. And we do see metabolic effects of spermidine in young mice on a high-fat diet and to some extent in older mice as well. So we're looking at this in more detail, and I think this data supports the idea that spermidine might be a beneficial longevity intervention to try in humans. So I want to come back to Singapore just in the last few minutes. And what I've been busy doing with a lot of help from colleagues here is to set up 2 centers in Singapore. One is a Center for Healthy Longevity that's focused primarily on human trials. And we have 3 planned human trials already, well, they would already be underway except for COVID-19. The first is a cross-sectional study looking at a range of aging biomarkers in the Singapore population. And then we're going to move into intervention studies. First, exercise, and second, we're going to do alpha-ketoglutarate, as I mentioned. And then we want to expand that to a range of other kinds of interventions and to be as agnostic as possible. So we'd like to try certainly some rapalog, but we're open to other people's suggestions as to things that might slow aging, and we will move these into clinical trials based on their safety profile and their ease of doing in human clinical studies as we have the resources to do it. We've also started a healthy longevity program at the NUS Medical School that's more basic research in nature, and we have a whole variety of themes now that we're targeting with regard to aging. And one thing I'll point out is that I think that what we're missing in part is that we just don't understand. We know aging is a risk factor for various diseases, but we really don't know why. What is it about aging that makes you more susceptible to neurodegeneration or strokes or sarcopenia? And so we want to try to dig into this in a little bit more detail to try to not just understand the disease and not just understand aging, but to understand the connectivity between aging and various diseases. And we have 31 faculty now participating in this program in various ways. And so there's a lot of research being produced. Now, what we really want to do in our translational approaches, though, is to combine two different things. One is state-of-the-art clinical studies on aging and frailty, and mix that with state-of-the-art preclinical studies on geroprotectors. And the way we want to do these preclinical studies is to match them as close as possible to how we're doing the clinical studies. So if we're using aging biomarkers in the human studies, we're now changing over to using aging biomarkers in shorter-term intervention in the mouse studies to try to be consistent. We can't look at survival in human studies, and so we need to move to aging biomarkers. And we think a number of these biomarkers are now at least validated enough that we believe in them, and we think they'll be very useful tools to understand which interventions are affecting aging. And then we're also using this frailty index that we have a lot of faith in. We have a number of studies also looking at mechanisms of various interventions. We have disease models that we're selectively choosing that will try to address this question of why aging mitigates disease. And I'm not going to talk about this today, but we're also trying to use AI to optimize the dosing of individual molecules in individual animals or in participants in the clinical studies. So if we can validate this approach in animal studies, we'll move it into human studies. So instead of giving everybody the same dose of a particular intervention, we will try to optimize that using artificial intelligence. And I can talk more about that if you're interested. So the clinical study, the primary thing we're looking at is healthy participants or relatively healthy participants, 45 to 65, relatively short-term studies looking if we can alter aging biomarkers. Now, I want to talk a little bit more about validating longevity interventions and what outcomes to use, because there have been a couple high-profile failures with interventions that have been developed because they target aging pathways. And those are largely in this first category, where you have a longevity intervention and you try to use it to treat some disease. And this approach has been taken by biotech and pharma companies for a long time. And there are good reasons for that, because if you want your drug to be approved by the FDA and get reimbursed, you have to be targeting an indication recognized by the FDA, and that's not aging, at least not yet. So the idea is to target a specific disease or some complication of a disease and that is recognized by the FDA, and then try to treat that with a longevity intervention. And I think we need to rethink this approach. If we really care about aging, we should be focused on aging directly and not trying to redirect our approach to specific diseases. Now, I get it. I know why these companies are doing this, and I'm not blaming them for it. At the same time, I'm afraid this is misleading us in terms of aging interventions that might actually affect health span and lifespan. And we need a better way of looking at that. So another way you could do it is to try to prevent disease or to prevent multiple diseases. The TAME study with metformin that Nir Barzilai is doing fits into this component very nicely. In a sense, if you're trying to prevent multiple diseases, you can argue that you're measuring health span. The challenge of this approach is that it's really expensive, and it's usually long-term trials. TAME is up to 3 years. So I really support this approach, but I think that it's not easy to scale that to look at lots of different kinds of interventions. And so I want to push a little bit this third approach, which is to look at aging biomarkers. And if you look in the literature, there's some very early studies, and many of them are not placebo controlled. They have small numbers of participants and other kinds of challenges. I don't deny that. But they're starting to show that some of the longevity interventions can slow or reverse the aging biomarkers. And I think this is a very fruitful area that we should be focusing on. One thing is this is amenable to lifestyle interventions and also natural products that don't have to go through full FDA approval. Now, these biomarkers have not been fully validated, so it's a bit early in that sense, but I think that these are, in my opinion, these are probably the best measures of biologic aging that are out there right now, and we should be exploiting them. And another reason I like that, and I'm running a little bit late, I realize, another reason I like that is that I think that the interventions were developed using one strategy. The biomarkers came from a completely different strategy. So it's not like everything came from the same approach. So if the aging interventions are really affecting the biomarkers, I think that's pretty good evidence that we're on the right track. Now, I won't spend any time on this. These are traditional biomarkers that have been used. And as you heard probably from Steve Horvath's talk, this is a review that we wrote. It's in Proteomics, if you want to find it. These AI-driven, omics-based molecular pattern biomarkers have really revolutionized the field, and they've been used to predict biologic age. And so the epigenetic clock, looking at DNA methylation patterns, is the most well-known, but there are clocks developed from these other pathways as well, and also others that I'm not even mentioning on this slide. And so the first generation of clocks were estimating biological age using a single data set. And now there are ones that are, I would say, second generation that combine single data sets with chronologic age and other clinical biomarkers like grim age. And I would argue that these are very effective for general population probably, but we need to get to this third generation, which is to really understand the disease-specific components of an individual so that we can understand how each person is aging in a personalized way. So looking at diseases is not sufficient. It may also be that looking only at biologic age is not sufficient. We have to combine these two things if we really want to get to personalized aging. And so we're using a wide range of parameters in our studies, including physiologic parameters like arterial stiffness, methylation age, facial pattern aging, aging data from complete blood counts in collaboration with Gero, and a range of other things to kind of combine and see how all these different biomarkers interact with each other. Do we need to measure everything to get your biologic age, or can we, how much redundancy is it in these different aging pathways? And this is really being developed by a research professor, Jorming Go at NUS. And then we're involved in a whole range of other clinical studies. I won't go into detail. There are longitudinal studies, other kinds of interventions that are being performed in Singapore. And we're trying to apply these aging biomarkers to a wide range of other outcomes, like predicting response to surgery in the elderly population, for instance. So all of this, I think, can be a huge benefit to Singapore, and it can be a huge benefit globally as well. It can benefit the individual by keeping them healthy, functional longer. This can extend extend productive work years to support the economy, which is a big issue in many countries. It's good for governments because they don't have to spend all their money building hospitals. As we've shown, we can save billions of dollars, Singapore dollars, by extending health span just modestly. And it's also good for companies that want to develop an industry. And so one of the things we're trying to do is bring companies into Singapore that are developing longevity interventions and try to help foster the longevity industry in this part of Asia. So I'll just end on this slide. And this is a book I read recently, just out of the blue, by Michael Pollan about psychedelics. Now, I'm not recommending psychedelics to slow aging, so do not quote me on that. But I thought the feature of the book was particularly interesting because, as you may know, psychedelics were widely used in research in the 50s and 60s. And then when LSD and other things entered the counterculture craze, the government got scared and made everything illegal, even though when they're administered in a way that's monitored, they were shown to be largely safe. And now in recent years, they've come back into the research realm, and there are a whole lot of potential benefits that are being studied. Treatment of alcoholism, treatment of depression, hospice care, um, depression in hospice care, and a range of other things. And the argument that the officials now are making, the powers that be, they're saying, all right, it's a good idea to test these things, but you should only treat the sick. You should not be working on the betterment of the well, even though there's a lot of evidence that some of these compounds may improve the life outlook for people that are not sick with any particular condition. And I think we have to get beyond this a little bit. This is sick care. We have to wait till somebody gets sick to give them something that might be beneficial. Now, I have no idea whether psychedelics will prove to be valuable or not in these contexts. That's not my point. But my point is that this is a widespread problem, and it applies to aging too. We need to stop focusing only on treating the sick. And these longevity interventions are not just going to prevent disease, but I think they're actually going to be involved in the betterment of the well. They're going to increase functional capacity in people and maintain functional capacity longer during their life. And so I want to get beyond this sick care mentality that is still pervasive throughout the healthcare industry and bring it back to healthcare, which is more preventative medicine. And I really think longevity interventions are one of the best ways to achieve that goal. So with that, I'll stop. I have a large lab now at NUS. The people here, Jorming has worked on the AKG project. I'm sorry, Shoping has worked on the AKG project. Jorming has done a lot of work on our clinical studies along with Jan. Yume and Chen Yu also worked on the AKG project at the Buck along with Garbo and collaborators Daniel and Azar. And Chen Yu is also working on the Spermidine project. I have a lot of collaborators and funding mechanisms, and thanks a lot for your time, and I'm happy to take questions. Sorry I ran over a little bit.
All right. Thank you very much, Dr. Kennedy. Those of you who are interested, I'm just going to remind everybody there's an on-demand talk by Thomas Weldon of Ponce de Leon Health, which was mentioned by Dr. Kennedy on our Lifespan Factory session, which is online, so do check that out. The questions. From the audience. So one question is, how do you see the big pharma model shift from treating disease to preventative medicine? And also, who are the biggest industry companies dealing with preventative medicine?
Yeah, I think it's very hard for pharma to shift because we have to realize that pharma is not losing money right now. A lot of pharma companies have, at least pre-COVID-19, were very successful by letting small biotech companies do the research, developing things to stage 2 trials, and then purchasing them and bringing them to market to treat disease indications. So if you're looking at pharma from a perspective of making profit, they don't really need our help. So I think that's one challenge. But on the other hand, I think that they're not recognizing the potential value of slowing aging. So they have a financial model, which is to make money off reimbursement for drugs. And I think that a pharmaceutical company that really has major resources that jumps into this game will find that there's a huge benefit to trying to prevent disease and keep people healthy and functional longer. It's a bit of a leap for them, but I think it's time for pharma to jump into this game. There are a lot of biotech companies that are making interesting progress in the aging space. I could list a number of them. I won't go into, you know them already probably. I'm very encouraged. I think there are all kinds of different ideas out there in this space, and I don't think we know which ones are going to work best in humans, so it's an exciting time. In terms of prevention, a lot of the companies that are very interested in that are nutrition companies or food companies, and so Nestle, Danone, and a variety of other companies are getting very interested in this space right now, and they may be very useful to help us develop natural products going forward.
So another question was, this is a bit more focused question on the data you presented. I believe this pertains to the animal data. How is frailty calculated? Somebody didn't quite understand that. And did you perform a dose response curve for alpha-ketoglutarate?
Actually, we're just now planning to do that in the human studies. We haven't looked at many different doses in the mice, and that's something we need to go back and do because it may be possible that we haven't achieved the optimal dose, and if we go higher, we could have an even bigger positive effect. We don't know that, of course. I will say that AKG is extremely safe. It's been used in human studies up to 6 grams a day for 6 months, in an osteoporosis study in postmenopausal women. And so we don't, that actually showed some efficacy, and that, so we don't really foresee any major safety problems. We're way under that dose in the product that's being marketed currently with PDL. But it may be that higher doses will have bigger benefits, and that's something we need to study.
I think, well, we have time for one more question. So another question is oral uptake and the effectiveness. So is oral uptake effective for both alpha-ketoglutarate and spermidine, and would it be more effective to combine uptake of AKG and spermidine? So I believe it's a bioavailability question.
Yeah, I think that they're both affected by oral uptake. But with AKG, we find that we use a sustained release format. So it's a lipid-based format that gets the molecule to the small intestine before it's released. Otherwise, I think a lot of it is probably consumed in the stomach. And the other thing we've noticed is that if you just take it on an empty stomach but you can buy the stuff off the internet that's just AKG without the sustained release version. And if you take it that way, it can cause a few minutes of heartburn because it's a very acidic molecule. So we prefer the sustained release format. The other question was combination of spermidine. That's something we need to do. We haven't done those studies, and I think that would be very interesting. I will point out that we've done a lot of combinations of natural products, and we can't predict necessarily the outcome. There are many cases where we put two things together that have benefits on their own, and they cancel the benefits of each other out. We never saw specific toxicity with any two of those molecules, but we did see one cancel out the benefits of the other. And so without testing spermidine and AKG directly, I wouldn't want to speculate as to what might happen.
Thank you very much, Dr. Kennedy.